Okay, then let's continue with the second half of neurocognitive methods, which is an introduction to the second very important technique for studying the human brain, namely fMRI. And this part of the course is really based, maybe even more so than the EEG was based on the LUC. This course is based on this book, Functional Magnetic Resonance Imaging, by Hüttel et al., which is really for a novice to the entire field of cognitive neuroimaging, a very good introduction to everything covering physics and the formation of images, which you mainly need in terms of theoretical background knowledge, but also covering then the biology and some cognitive aspects of this in terms of experimental design, and also covering data analyses for fMRI data at a level that serves as an introduction. It doesn't serve, in terms of data analysis, not really much more. I mean, the whole book doesn't serve much more than an introduction, but it actually delivers on the introduction quite well. So if you start from zero, this is where you should start. But if after you read that, you should not stop reading, but you should then really start to read the real stuff. But it's a very good book. And the figures are all from that book, essentially. So I would really recommend, if you want to work in cognitive neuroimaging, buy this book, read it, and then read more. This we've already seen, so we don't have to discuss much of this again. So we are basically now changing a little bit the temporal resolution, maybe going to a little bit of higher spatial resolution, while looking at lower temporal resolution. But yeah, we are still somewhere here in terms of trying to resolve neural activity in the order of seconds to milliseconds with the ERP, and at the order of centimeters to millimeters at the level of Dreyri and Salzai. This is where cognitive neuroscience happens, after all. So that's the outline for the course. It's fairly similar to for this part of the course, what we've seen in EEG. So we start again with an overview, an introduction that covers basically everything in a nutshell, so to have some basic concepts established. And after that, we talk a bit about the physics of the imaging process and the signal processing involved in the imaging process. After that, we talk about the basic biological model that is thought to induce these signal changes that can be observed with the help of an MRI machine, which here comes under the label Bolt signal. So that Bolt signal is essentially the dipole model. So it doesn't have to do anything with dipoles, but in terms of its function, it's kind of the same model type that as the dipole model served in EEG. Then we talk about some aspects of the Bolt signal, essentially what one can do and what one cannot do with the Bolt signal. A bit on experimental design, which is just essentially some nomenclature, and then ending up here in terms of fMRI data pre-processing, so discussing a typical pipeline of pre-processing steps. So it's again a little bit like with EEG. There are some conventional things that most people do when analyzing fMRI data. So like most people use some kind of eye blink correction in EEG. Similar kind of things in fMRI. And then I think we, and especially you, are in a good position to next week start learning about data analysis. So one idea about this whole doing cognitive methods before we start with the statistical methods this year is, of course, also that you have some background content and context for what the data analysis techniques that we then discuss actually build on. So that next Monday, when I show you what we will do in statistical methods all year in terms of, not all year, but in the winter term, in terms of data analysis, then you know from this week where this actually starts from. So that's the plan. Any issues with fMRI you already have, or that you want to bring up? Anything before we start? No, I guess not. Why should there be? Starting, as I said, with a kind of quick run through the whole MRI/fMRI business. Essentially in kind of this next hour or so, briefly introducing the physical foundations, the biological foundations, and some statistical foundations of data analysis. Again, like for EEG, as a cognitive neuroscientist, you will worry about the physical foundations and the biological foundations not on a day-to-day basis. On a day-to-day basis, you are occupied with either acquiring or analyzing these data, as for EEG. But of course, when reading papers, you come across terms that relate to the physical foundation, so things like TRs or something. And of course, it's always good to keep in mind that the actual origins and biological foundations of EEG signals, fMRI signals, are understood to a certain degree, but not fully, and that this is actually an ongoing kind of research endeavor, and that one should use, or that one should use some modesty when making now biological claims about the foundations of cognition when using these techniques, and always be modest and not overstress things, and write papers that have the title "How the Brain Does This and That," because the article will not answer that. And that's good for to know about the biological foundations. And we step right in. So these first couple of slides in terms of the physical foundations deal with the name. So I guess you all know that these machines are called MRI scanners, so magnetic resonance imaging scanners, and the whole technique is referred to as magnetic resonance imaging, and functional magnetic resonance imaging is a specific form of magnetic resonance imaging. And and the question that is addressed in these first couple of slides is what is the magnetic aspect, what is the resonance aspect, and what is the imaging aspect, and also why the name. And the first thing is the magnetic part in it. As you, I guess, know, these magnetic resonance imaging devices that are, of course, also available in hospitals, and actually that's their main function. Their main function is not to do fMRI experiments, but their main function is to do clinical diagnostics with. They are based on very strong magnetic fields. So as you, of course, all know, this is an imaging process that doesn't involve any form of radiation, like computer tomography or X-rays, but involves strong magnetic fields. And hence the term magnetic in it. These machines, they comprise a very strong static magnetic field, and then they also include a number of what is referred to as coils, namely radio frequency coils, gradient coils, and shimming coils. Radio frequency coils basically sending electromagnetic waves that interact with the atomic parts of samples in this static magnetic field to support the imaging process. Gradient coils that establish certain spatial variations of the magnetic field at given time points, which are necessary for the imaging process. Shimming coils that contribute to having a very homogeneous static magnetic field where any gradients are not random but are fully specified. And then of course all of this, so this kind of machine is accompanied by a lot of computer hardware and software. So this here is a GE scanner. Over at the CCNB, the scanning room actually looks quite similar, but we have a Siemens scanner here. And I guess you've seen this before. So who has been inside an MR scanner? Can you raise a little bit more? So I get an impression, so like 50%. Everyone else who is interested in this, I would really recommend taking part in an fMRI study that is conducted here. Then you get first-hand experience of how it is to be a participant in such a study, and you also get to see the scanner and the interior of the scanner and so on. So you see advertisements for these studies. So there are some, I guess you've seen them around, and there are always studies being conducted. You get a little bit of money, and then of course the people who you interact with there are very happy to also explain what they're doing. It's usually PhD students together with some other people who will scan you, and then you learn something, and it's worthwhile if you do it once, twice. At one point of course it gets boring, but at least going to it once is definitely a good idea, so that you see it from a first-hand perspective. The magnetic part, a little bit more on this. There is the static magnetic field, which is created by the scanner, and which you can actually detect if you go into this room. So it actually draws magnetic things onto it. Is referred to as B0. So that's just a term you can learn about. This magnetic field is created by a electromagnet. This electromagnet means that there's some current that induces this magnetic field, and it's superconducting in the sense that this whole thing is this coil that generates the magnetic field is cooled down a lot. So this is why this whole imaging process needs helium as a cooling agent, and this cooling is required to get the resistance very low, so that you basically once have to charge up the scanner, and then there's no need for current supply all the time. There's only need for helium supply. So the power requirement is quite low, but the helium requirement is not that low. You will have heard already last Wednesday when Rainer Göbel gave his talk that there are and have been scanners of different magnetic field power, or strength, I should say, different strengths. So in the 90s, 1.5 Tesla scanner were kind of the norm. Then in the 2000s, and actually until now, 3 Tesla scanners are the norm. So in terms of getting an intuition, so these electromagnets, they are as strong as these electromagnets that you maybe have seen sometimes at a car graveyard, is that a word? Schrottplatz. That can lift cars essentially by using some magnets. So it's kind of this kind of strength of magnetism that is involved here. Field strength can help because this is quite a complex setup, as we will see over the next couple of hours. Field strength can help with the spatial resolution, so you can have higher spatial resolution, or you can have quicker imaging time. It's not that everything just gets better if you have higher field strength, because there are also biological limitations to the whole thing. So currently the standard way to do imaging is with 3 Tesla scanners. Higher Tesla scanners, like 7 Tesla scanners for humans, are available, but they are a little bit more on the experimental side. So the typical clinical scanners will be 3 Tesla scanners, which have been validated by the companies and so on quite a lot. But there are also 7 Tesla scanners. And what is quite important in terms of kind of the physical maintenance of scanners is that the field remains uniform, and hence additional coils, and it's just important for the imaging process. So just as a first, we will go over details again to some degree. So just that you know that there's something called B0. It needs helium, and it doesn't require a lot of power. So why then resonance? Resonance relates to the fact that there are these radio frequency coils that excite atomic nuclei. So radio frequency coils basically being just something like in your microwave at home. I don't know whether people still have microwaves, but who owns a microwave? That's what I guess. People usually don't. Well, do people use microwaves still? I don't know. I haven't. So since I left my parents' house, I didn't have a microwave.
Yeah, in the States they're really common. I was actually surprised that people don't use them here.
Okay.
I use it every day.
You should think about what you eat, or if you use it for something, you can also of course use it to cook water. But there are these books like I don't know whether it's a joke, but I think it's real. It's like microwave diet for the single man or something. There are these books from the 70s or 80s. Anyway, I don't think with the whole organic food, bio stuff and so on, that microwaves are that popular anymore. Anyway, but these microwaves, they also emit radio or electromagnetic waves to heat stuff, and kind of the same thing is built into a scanner, but of course not with the aim of heating things, but at a lower kind of power to basically interact with atomic nuclei. And then the thing is that based on some excitation of atomic nuclei, which we'll look at in more detail later, these atomic nuclei also emit electromagnetic power, and this is again picked up by the radio frequency coils in terms of antennas. So if you think about a radio that has an antenna, it picks up the electromagnetic waves floating through the air, and you can hear something. This is also happening here after some energy has been transferred to the sample. It's emitted again, and based on the characteristics of this emission, the imaging process then happens. So there's some reception, there's a reception of atomic nuclei radio frequency energy. The coils, these radio frequency coils, come in different forms, so one can functionally characterize them as transmit and receive coils. So these would be radio frequency coils that send out some electromagnetic waves and also act as antennas, but there are also coils that are only to transmit, so induce radio frequency waves, or to receive data. So for example, what you've seen here, there will also be pictures on the next slide, but here so for example, the Philips system that we had in Birmingham, or still is in Birmingham, there was a body coil built into this what is called the bore of the scanner, which emits radio frequency and also records radio frequency from the sample, whereas the head coil that is typically used for brain imaging was only a receiving coil. So the setup used the radio frequency pulse and excitation from the body coil, and for having better imaging properties of the brain that we are interested in, there's then a pure reception head coil. So they come in different functional types, and they come in different geometric types. So the typical thing these days is actually to use what I call phased array coils. That have good sensitivity and a good coverage. If you haven't seen that lately much, but if you're, for example, just interested in visual cortex and you are really interested in getting good signal from visual cortex and not the rest of the brain, you can also use these surface coils for reception. These volume coils or birdcage coils is kind of a little bit more old school way to image. So the typical coils that are used these days look like this and are called phased array coils. So this is basically that you know how different coils look. In addition to these radio frequency coils that send signals and energy to the sample that you're trying to image and receive it, there are gradient and shimming coils. The gradient coils essentially adding gradients to the magnetic field. So basically in a controlled manner, make sure that the magnetic field changes over distance, and they are really important for the imaging process. So we will actually see them again when we talk about Fourier imaging. In addition, there are shimming coils that try to, if there are some inhomogeneities induced in the magnetic field, for example, due to the sample that is placed, so the human that is placed into the magnetic fields, the shimming coils basically try to undo these inhomogeneities. And this when you run sequences, it's called, so when you run imaging procedures on the MR scanner, there's usually a shimming part where you hear, because all of this makes some noise, where you hear the shimming coils being adjusted as a kind of characteristic noise. And you also don't get images, and then people would say, this is just the shimming, and okay, that is there to make the magnetic field homogeneous, because that's important for the imaging process. And this is what I just explained. So the entire setup is then, of course, not only the scanner with its different coils and its electromagnet as it's in the room, but there's some technical driving material behind it. So there are amplifiers for the gradients, so that the gradients can be turned on and off. There is the radio frequency amplifier and transmitter, maybe a pre-amplifier. This whole thing, of course, again, needs a process that converts digital information into continuous physical information, and the other way around. So there might be something like a digitizer. And of course, all of this is controlled by a computer. So essentially, there's a scanner control console where you have a graphical user interface and can turn on the imaging process and turn off and say how the imaging process should be done. In addition, in these kind of setups, you of course have, again, in a cognitive neuroscience context, stimulus PCs and, for example, certain experimental devices that people in the scanner can use to take part in some cognitive tasks, like pressing a button or using a joystick. Or what also, of course, is often present in fMRI setups is some mirror, so that people can see a screen where with a beamer you project your experiment, if it's a visual experiment, or people put on some goggles on which you present your experiment. So this is kind of the entire setup. The participant usually only sees this and a little bit of this when they come in. You are mainly dealing with this, and the technicians and essentially technicians of the MR suit then also deal with this part. Actually, the way this really works is if the scanner is not working. So the way MRI actually works at a university is that there's the MR center. You need to buy the scanner, and you remember from last Wednesday there were some pictures of when the scanner arrived. And then there's a contract with the company, in this case Siemens, who basically do the maintenance. So whenever something is not really working and you get an error message, it's not that there is somebody around who will now then go and say, okay, and then they will open here then Felix or me or somebody will come and open the scanner. But it's essentially, and also not the radiographer like Christian Kainz, but Siemens will be called. Actually, they get an automatic because the system is obviously connected to the internet, and they get an automatic notification that something is not working, and then they will take care of it. And this whole maintenance contract is 250,000 euros a year. So the task for the scientists is to ask the third-party funding people for money to run studies, so that this money that they then get from like a research foundation can be transferred to Siemens, so that we can pay for the maintenance. That's how this is kind of how the research or the academic system contributes to economic growth in Germany, by basically using state money from everyone to give it to the company. And our task is to come up with reasons, scientific reasons, why we want to give the money to Siemens and the shareholders of Siemens. Yeah, so everybody pays, and we give it to the shareholders of Siemens. That's the system. And it's actually the system. So that's 250,000 a year, which is money that you don't spend on EEG, for example. So there's no maintenance contract for EEG systems. Of course, you have some guarantees, and they then repair your EEG system, but you don't have to pay just to have the machine there running. So one thing that is important about magnetic resonance imaging is that, also in contrast to EEG, is that it's dangerous. And a lot of care needs to be taken that nobody gets hurt. And this is actually really important. So the magnet is always on. And if care is not used in dealing with the scanner, then things can go wrong. This shows two examples of kind of a hospital bed being drawn to the scanner because of the magnetic field, and here kind of a medical device. Hence, one needs to be you really need to be on your toes when and be careful when you use magnetic resonance recording for your research. You can also, of course, not just scan any participant. There are contraindications for scanning things like metal implants, which, if they are in the human body, they can, of course, also move, or at least move, and then induce some internal damage without the need for a human being kind of between this bed and the scanner, which is of course also very damaging. Things like pregnancy, although the bottom line in the story is there is no problem at all during pregnancy for cognitive experiments. There's also no scanning, and so this is important. And when you do MRI experiments, there are screening forms where you make sure that your participants don't fulfill this contraindications. And whenever you enter the scanning room, you have to make sure that you don't have any metal on you, so that the metal doesn't fly through the room towards the scanner and may hurt someone in the way. And you also have to make sure that the participants don't have any metal on them. yeah, and this is really important because there's completely no reason that anyone should get harmed for cognitive neuroscience, obviously. So this is just important, and there are yearly safety sessions. I think there will be one, and this will, I think, also be announced again in the third week of the term. So not next Monday, but the Monday 2 weeks after, before the CCNB seminar, because everybody who wants to work on the MR scanner needs to take part in these kind of safety briefings once a year, and it's important because things can go wrong and have gone wrong. So that's really something to be really clear about and be aware of. And if you don't like this potential danger that you expose yourself or participants to, you can also just not work with MRI and do EG. Then you don't have to worry about this. But if you work with this, you have to worry about it. Yeah. So that's a little bit on the physical side. So basically there's magnetic resonance and imaging, and the imaging part here relating really to the gradient coils. Now we will talk about this in more depth. Strangely enough, in these introductions to fMRI, I always find there's a little bit more of an emphasis on the physics than there is an emphasis on the physics in EEG. I think it has to do with the fact that it's more fascinating how magnetic fields can create images, whereas that some electrical activity can be recorded, although I have also no idea how that works. It doesn't seem to be that fascinating, so we will talk a little bit more about the physics than in EEG. But now, first, something about the biological foundations, and this is essentially the same stuff that Rainer Göbel mentioned last week when he gave this little introduction for a general audience. So the most important part for fMRI in terms of imaging is the fact that the bold signal is related to the metabolic demands of active neurons, where bold stands for blood oxygen level dependent signal. So somehow the requirement of neurons to eat oxygen can be detected in fMRI in a spatially and temporally specific manner. So very roughly. So the bottom line here is, this is one slide trying to introduce the bold signal. So the bottom line is that, of course, the brain needs energy to maintain and restore membrane potentials. As you remember from EEG, there are things like membrane potentials, and they need to be maintained, sodium-potassium pump, and restored after excitation. However, the brain has no energy stores per se, so energy must continuously be provided. So energy stores in your body are, for example, the glycogen in your muscles and your liver, or your the fat stores that can be used to create energy. But the brain, so the neurons, they don't have like glycogen stores or fat stores, so the energy must always be provided in terms of the blood stream. And of course, the main energy source the main energy sources for the brain are glucose and oxygen. And they are used to create ATP, which is of course used in terms of the sodium-potassium pump. The oxygen is delivered via this blood protein hemoglobin, which is part of your red blood cells, and then the for two wishes, the The good thing is that oxygenated hemoglobin, so hemoglobin that binds to oxygen, has different magnetic properties as compared to deoxygenated hemoglobin. So if there's a change in the level of oxygenated versus deoxygenated hemoglobin, this leads to local variations in magnetic properties, and these properties actually then induce differences in the MRI imaging process that can be detected. So this is just one slide that is more, again, for you to, when you study this, memorize kind of the sequence. We will talk about this process multiple times, and then you will become more familiar with it. This is also something Hauke mentioned last Wednesday. I don't remember the context, but it was something about 1992 and the first imaging fMRI experiments in humans. So these are these papers from 1992, where for the first time human participants were imaged in an fMRI setting. Of course, MRI had been around by that time for longer, but based on new sequence design and some findings in animals, they had the idea that this should also work in humans. And what happened there is that they presented visual stimuli to participants and looked at the signal intensity in some visual cortex area, and they report that the signal intensity increases. And same thing was then also reported for motor activity, for motor cortex. And there's another kind of classic study that again uses some visual stimulation. And this is also something, so these kind of effects that if there are human participants in the scanner, then the MR signal in visual cortex increases. This is really robust. I mean, this is basically reproduced every day in cognitive neuroimaging when we show images and visual stimuli to participants. It's also in a way where the whole thing comes from. So it was, you need to keep in mind that it was not that there was first somebody dealing with neurons and then somebody dealt with some blood activity, and then later somebody said, okay, let's do so that this entire setup was kind of built up in a designed fashion. The basic finding is the signal that you can record increases under visual stimulation, and then the question is why. And so this process from neural activation to changes in MR signal was not designed by anyone or something. It's an empirical fact, and then everything that in terms of the theory tries to explain this is kind of a model and explanation for this finding. Yeah. So just that, because that is sometimes, so the basic fact is the MR signal goes up if there is if people are exposed to visual stimulation. Then the question is why. And this again shows kind of the entire pathway of the increases in MR signal intensity, which we'll see again, but we see it here as well. So if you have in some brain area an increase in neural activity, the question is then what kind of neural activity exactly, and we'll see something about this. Then what happens is that this area increases its oxygen consumption, but it increases its cerebral blood flow more than it does the increase its extraction of oxygen. This then leads to the fact that actually when there's activity, there's an increase of oxyhemoglobin and a decrease of deoxyhemoglobin, which is a little bit counterintuitive because you would think if there's activity, the oxygen is eaten up. But actually there's an overcompensation in terms of the cerebral blood flow, and you have an increase in oxyhemoglobin. And this leads to an increase in the magnetic susceptibility of this area, leading to an increase of a time constant in MR imaging, and this then leads to an increase in the MRI signal intensity. So we will come back to this, but as a kind of pathway and also as a help for memorization, this is of course helpful. Yeah. So finally, then maybe in the last part of this hour, something about fMRI data analysis, because again, this and the stuff that we saw in the physics before, you will not really deal with if you're not doing research on it. So if you want to do MR physics or develop new sequences, feel free to. If you want to study the biological underpinnings of the Bolt signal, also feel free to go to a lab that does that. But if you do anything else in terms of cognitive neuroscience, you will mainly be dealing with making sense of observed fMRI data if you're using fMRI. And so this is really what you're dealing with a lot. Actually, no, I think I want to take a break before we do that, because it's important and I want us to be a little bit fresher before going into this. Any questions so far? No. Good.